Good afternoon. Thank you very much for being here. I'm going to be presenting today some more data about the link between energy homeostasis, neurodegeneration, and aging. So let me begin by the question that we are trying to address. Is it? Okay, I have to go one. So this is a very simple question, actually, but it remains a mystery to a very large extent. We would like to understand how neurons, specifically neurons, among all cell types, maintain their homeostasis over very long periods of time. The reason being that neurons are the most long-lived cells of our bodies, as you've heard this morning. They face unique challenges. Not only do they need a lot of energy to function throughout their life, just sitting there idling, actually neurons consume energy, even just doing nothing, just sitting there, because they need to maintain a polarized plasma membrane to be able to transmit electrical signals. In addition, they have a unique architecture. As you've seen here, they have appendages, very long axons, dendrites, synapses, etc. And it costs to develop, establish, and maintain this complex architecture. And of course, they are complex cells and energy-consuming cells. So in order to do that, they have to produce a lot of energy, and they do that in their mitochondria. Mitochondria are the subcellular organelles that are generating energy in our cells. In neurons, mitochondria are specifically important because neurons cannot produce energy, for example, by performing glycolysis. That is very common in our muscle cells. So mostly they rely on oxidative phosphorylation, and this is what's going on in mitochondria, the respiratory chain, and that's how they produce their energy. So if something goes wrong with mitochondria, we have profound neuronal pathology. In this case, I'm showing you the accumulation of mitochondria in Alzheimer's disease patients. This becomes a common denominator of many neurodegenerative disorders, the accumulation of damaged mitochondria. So we wanted to understand how neurons actually make it, how neurons can establish and maintain homeostasis throughout tens of years, even more than 100 years on occasion. And this is actually an interesting question in the context of aging as well, because we see that there is accumulation of mitochondria in various tissues, including neurons, during aging. In this case, we are comparing rats and C. elegans, and there is an accumulation of mitochondria as these organisms age. So this appears to be a feature of the aging process itself. And what made it even more interesting for us, and this was one of the reasons we decided to investigate further, was the following observation that was made some years ago in the lab by a talented postdoc. So what Marta Artal observed was that while in wild-type animals, we have this accumulation of mitochondria over time during aging, in long-lived mutants, C. elegans strains that live longer, we actually don't get this accumulation. So this links accumulation with aging, or accumulation of mitochondria with aging itself. It could be that there is a causative relationship there. Now, in order to be able to establish a causative relationship, because at this point, this is just a correlation. Long-lived animals don't accumulate mitochondria, whereas normally, normal animals, wild-type animals, accumulate their mitochondria. We would need to have a way to actually influence the number of mitochondria in cells on purpose, and then find out what's going on with lifespan. And this is exactly what we decided to test. So I'm not going to go into the details how this experiment was done. Suffice it to say that there is a way to actually genetically dial in the number of mitochondria in C. elegans in nematode cells. So we can, for example, mutate certain genes encoding proteins called prohibitins. And in this case, if we mutate these genes, we exacerbate accumulation of mitochondria in wild-type animals. However, if we do the exact same thing in DAF2 mutants, long-lived mutants, these are, by the way, insulin-defective mutants, that's why they live longer, we see the exact opposite effect. These animals now accumulate even less mitochondria over time. So what is happening during aging then? We see that when we exacerbate the phenomenon of mitochondrial accumulation, there is a shortening of lifespan. But when we attenuate mitochondrial accumulation, we actually extend lifespan even further. So it seems that enhancing accumulation of mitochondria, this is the conclusion from these experiments, enhancing accumulation of mitochondria shortens lifespan. So having more mitochondria accumulating in cells is not actually good for you. Whereas if we block accumulation of mitochondria that happens during aging, if we find ways to attenuate it, then we have longevity. We live longer, at least in this organism. Of course, these experiments have now been repeated in Drosophila and in mice, and we see the same effects. So we decided to address this question. How do we lose control over the number of mitochondria in cells? How do we get this runaway accumulation of mitochondria during aging? Usually what happens is that we have two processes balancing each other to determine the number of mitochondria in cells. Mitochondrial biogenesis and mitochondrial degradation. Very simple. However, it appears that during aging, or in the context of age-associated pathologies, this balance is lost. And in turn, we end up with runaway accumulation of mitochondria. So we did a genetic screening in C. elegans, and the way we did the screening was actually to use the property of long-lived mutants that don't accumulate the mitochondria. So we have ways to stain mitochondria. We have ways, dyes, specific dyes. These are called mitotracker dyes that can be used to stain mitochondria red. So we can visualize mitochondria in animals as red dots inside cells. So the more mitochondria you have, the more red you become. And if mutants are not accumulating mitochondria, then they are darker than wild-type animals that accumulate mitochondria. So the hypothesis here, the working hypothesis that we made was the following. We hypothesized that in these animals that don't accumulate mitochondria, homeostatic mechanisms are actually at operation in a more efficient level. So they can cope with this accumulation of mitochondria, not allowing it to happen. That's why they are darker. So what if we go in with a mutagen and we hit those genes that are participating in the relevant mechanisms? We would revert then back to wild-type conditions. We would now allow mitochondria to accumulate. And this is a very convenient way of doing the screening because we can look for brightly red animals and identify them very easily in a sea of dark animals. So this allows us to screen very large numbers, and as we call it in genetics, saturate the genetic screening to identify all genes involved in a specific process. And we did exactly that. I'm going to tell you today about 3 genes that we recovered from this genetic screening. These genes are called DCT1. It's actually the homologue of NIX in humans. NIX, DCT1 is a mitophagy receptor. I'm going to explain a little bit later. PINK1 is a serine threonine kinase, and parking is an E3 ubiquitin ligase. The name parking refers to the Parkinson's disease because this protein was actually isolated and studied in the context of Parkinson's disease before. So now these 3 genes are involved in a process called mitophagy, and we described it this morning, so I'm not going to go again into the details. But I'm going to tell you now a little bit more about the pink parking pathway for regulating mitophagy. And this is the last paradigm of mitophagy shown here. So what happens when cells sense that there is a damaged mitochondrion around? What we see happening is that there is local activation of the PINK1 serine threonine kinase. This happens because of a mechanism that involves ROS that are generated by that particular mitochondrion. So ROS generated by the damaged mitochondrion activates PINK1 in the vicinity of that damaged mitochondrion. Then this, in turn, activates parking, phosphorylates and activates parking, the E3 ubiquitin ligase, which then starts ubiquitinating specific proteins on the outer membrane of mitochondria. And this serves as a flag signal for the autophagosome to be created around the damaged mitochondrion. So this is the process of mitophagy that's been induced by pathology. In this case, pathology is the appearance of a damaged mitochondrion. Now, in C. elegans, this pathway operates similarly to humans. So we have a sequential activation, pink 1, parking. Then we have ubiquitination of DCT1. DCT1 is a mitophagy receptor, as I told you. Is a protein on the outer membrane of mitochondria that becomes ubiquitinated, and specifically recruits the autophagosome to be formed around the damaged mitochondrion. So we were able to identify even the lysine residue that becomes ubiquitinated by parking upon induction of mitophagy. Now, if mitophagy cannot proceed, and now we have a way, and this is what a mitophagy receptor allows us to do. It allows us to manipulate mitophagy specifically away from other types of autophagy. So we don't want to interfere with general autophagy. We don't want to interfere with other types of autophagy, just interfere with mitophagy. And we can do this if we have at hand a mitophagy receptor. And in this case, we can knock down DCT1 and see what happens to lifespan of long-lived animals, for example, DAF2 mutants in this case. So what we see is that we take away part of their longevity. This suggests that these animals, which are insulin signaling deficient, actually live longer because one of the reasons is that they activate mitophagy. And if we now bring it down by removing an essential mitophagy receptor, we shorten the lifespan of these animals. Now, in wild-type animals, if we do the exact same thing, if we knock down a mitophagy receptor, DCT1, in wild-type animals, we don't see a difference. In this case, of course, one might expect that because wild-type animals don't have activated mitophagy. That's why they don't live as long as DAF2 animals. So essentially, if there is no mitophagy, substantial mitophagy happening in wild-type animals, actually removing a mediator of mitophagy would not make much of a difference. However, this generates a paradox, and I'm going to tell you a little bit more about that in a while, why this is a paradox. But for now, suffice it to say that mitophagy activation contributes to longevity in long-lived C. elegans mutants. And these experiments, again, have been confirmed in mammals. Now, in the case where we downregulate mitophagy, not only do we compromise longevity in long-lived mutants, we also now allow for accumulation of mitochondria, especially in neurons. Neurons are particularly sensitive because they rely on mitochondria, so they start accumulating mitochondria upon downregulation of mitophagy. And when this happens, we have a phenomenon of oxidative stress ensuing inside neurons. Why? Because now we accumulate damaged mitochondria. These are not removed, as they should, through mitophagy. So they remain there, they start accumulating, and damaged mitochondria produce a lot of reactive oxygen species. They leak calcium. Mitochondria are the second largest calcium reservoir in cells. There is a collapse of the inner mitochondrial membrane potential that is used to produce ATP, and as a result, energy production becomes lower. So we have a phenomenon of mitochondrial aging. We start with young, functioning, fresh mitochondria. We have a lot of ATP being produced, very little ROS. Aged mitochondria, damaged mitochondria, a lot of ROS, very little ATP. And as a result, these cells, these animals, experience chronic oxidative stress if we downregulate mitophagy. Now, because of that, we now face a paradox. If we have animals that are faced with chronic oxidative stress, we would expect, at least, that there would be shortening in the lifespan of these animals, because oxidative stress is detrimental, generally, to organisms and cells. However, we see that if in wild-type animals we downregulate mitophagy, and in this case we use all three genes involved in mitophagy to downregulate mitophagy, lifespan remains steady as in wild-type animals. And this is a paradox because lifespan is the most sensitive gauge that you can use to monitor anything that's going wrong with a cell. Because it's a phenotype that develops over time, so there are cumulative effects. Anything, even the slightest effect, the slightest intervention will have a measurable influence on lifespan. And in this case, we don't see it. So why is this? So we thought that maybe there is a way compensating, there is a mechanism that compensates for the downregulation of mitophagy. And this mechanism could be retrograde signaling. Now, what is retrograde signaling? Actually, mitochondria themselves have their own DNA. They actually originate from bacteria that invaded cells in ancient times about 2 billion years ago, and that's how we now have mitochondria. But most of the genes of mitochondria have now migrated to the nucleus. So mitochondrial proteins are encoded in the nucleus, and there is a transcriptional program that regulates the expression of mitochondrial genes in the nucleus. This transcriptional program is orchestrated by PGC1 alpha. This is a master transcriptional regulator that works together with cofactors of the NRF family of cofactors to upregulate the transcription of mitochondrial genes whenever cells sense that there is something wrong with their mitochondria, that they need to build fresh functioning mitochondria. And this retrograde signaling involves calcium, mostly, that leaks out of damaged mitochondria. That's how cells realize that they contain damaged mitochondria, because of the calcium that leaks out of mitochondria, but also because of ROS. Now, in C. elegans, we don't have a PGC1 alpha if we want to approach this pathway, but we do have a transcription factor of the NRF family called SKIN1, and we decided to look into this further by looking what happens to SKIN1 and the activation of mitochondrial and mitochondrial genes in C. elegans. So indeed, we find that upon downregulation of mitophagy, the cells now activate retrograde signaling by activating this SKIN1. SKIN1 is a transcription factor, which in turn activates the transcription of mitochondrial proteins, mitochondrial protein genes. And these are just some examples of such genes of the respiratory chain. But interestingly, among them is DCT1 itself. Now, DCT1 is a mitophagy mediator. So it's as if the cell is trying to do two things. On one hand, to build new mitochondria, upregulate the expression of genes encoding mitochondrial proteins, but at the same time also induce mitophagy to take care of the damaged mitochondria that have been accumulated. And it does so by putting DCT1 under the control of a master regulator that also controls biogenesis of mitochondria. So if SKIN1 is not there to compensate, to provide for this compensatory mechanism, then downregulation of mitophagy should have a strong detrimental effect on lifespan. And indeed, that's what we see. In the absence of SKIN1 that would restore a biogenesis loop, we now have shortening of lifespan upon downregulation of mitophagy. Now, going back to neurons, and this is the last part I'm going to tell you today, what we see happening, if neurons experience oxidative stress, they sort their damaged mitochondria and actually transport them to the soma, to the neuronal soma. That's where most of the degradative machinery resides, the lysosomal system that eventually will degrade these mitochondria. So they will need to transport their mitochondria from long distances, sometimes meters away. Some of our axons in our bodies are more than 1 meter away. They will need to transport their mitochondria to the soma of the neuron for degradation. And this is happening upon oxidative stress. So first thing, sort damaged mitochondria and transport them to the soma. Second thing they need to do is they need to upregulate mitophagy. So upon oxidative stress, what we see is that mitophagy is upregulated. Mitophagy needs to take care of damaged mitochondria. So neurons naturally upregulate mitophagy upon oxidative stress. Now, if we intervene genetically, not allowing mitophagy to become upregulated, then of course we have accumulation of damaged mitochondria. Even the sorting mechanism now doesn't work well because we have accumulation in axons, dendrites, and other neuronal appendages of damaged mitochondria. And as a result, we have sensitization to neurodegeneration. In this case, we can, for example, establish a C. elegans Parkinson's disease model. How? By overexpressing human alpha-synuclein. It's an aggregation-prone protein that has been associated with neurodegeneration in Parkinson's disease. If we overexpress human alpha-synuclein in dopaminergic neurons of C. elegans, because those are the neurons that degenerate in Parkinson's disease, we see that there is the neurodegeneration. We can even fine-tune the levels, so we get about 10% neurodegeneration. Now, if on top of that, we downregulate mitophagy, these neurons become extremely vulnerable to neurodegeneration. We have a sensitized neuron now, a sensitized system, and these neurons degenerate a lot more. That's why we see much less survival. So these are surviving neurons, less and less surviving neurons. And in another model of neurodegenerative disease, in Alzheimer's disease, in this case, we overexpress tau and beta amyloid. Those are, again, two aggregate-prone proteins that have been associated with Alzheimer's disease. And we see that there is strong inhibition of mitophagy upon downregulation of, upon overexpression of tau and beta amyloid. There is strong downregulation of mitophagy. So this is actually a mechanism that contributes to pathogenesis in Alzheimer's disease. And we've confirmed it in post-mortem brains of Alzheimer's disease patients. These patients do show downregulated mitophagy in their neurons. Now, I've told you what mitophagy does in a pathological context in two neurodegenerative disease models. What about the normal function of mitophagy in physiological neurons when we don't have insults? In this case, we looked at the contribution of mitophagy in physiological neuronal functions, such as, for example, learning and memory. Now, these animals, simple as they might be, worms, actually can learn, and we can teach them things. How? Using the old Pavlovian way of learning, associative learning. So in this case, what we do is we pair, for example, a chemical smell, such as isoamyl alcohol, with starvation. Usually these worms, innately, they like chemotaxing towards this compound. To them, it signals the presence of food, because in the environment, these animals eat bacteria, and isoamyl alcohol is a product of fermentation caused by bacteria. So nature has made it so that their nervous system is hardwired to like isoamyl alcohol. That's why animals that are not trained very strongly chemotax towards isoamyl alcohol. However, if we train them by pairing a chemoattractant, isoamyl alcohol, with a condition that they don't like, such as starvation, so we punish them by putting them on an empty plate with isoamyl alcohol, they now learn not to like isoamyl alcohol. So chemotaxis index, instead of being positive, is now negative, which means aversion. Not only they don't like it, but they don't even approach it. So we have behavioral plasticity that signals learning and memory. So they've learned not to like isoamyl alcohol. However, if we now interfere with mitophagy, we do see that there is behavioral plasticity, but they never learn to avoid isoamyl alcohol. They still like it, they never learn to avoid it, which means that basal mitophagy in neurons serves a homeostatic function that is important for processes such as learning and memory. So I'll summarize now what I've told you today. This is a simplistic scheme that actually also, I think, demonstrates the economy of nature. Having one mechanism actually regulate two opposing processes. On one hand, mitochondrial biogenesis, which creates new mitochondria. On the other hand, mitophagy that turns over mitochondria. How is this coordinated? Whenever cells sense that there are damaged or superfluous mitochondria, two things happen. On one hand, we have oxidative stress, as I've told you, and oxidative stress through a process of signaling. I didn't tell you details, but this signaling involves many proteins, different kinases. Eventually, we have activation of a master transcriptional regulator, SKIN1. Now, SKIN1 does two things. On one hand, induces mitochondrial biogenesis. On the other hand, it induces the expression of an essential mitophagy mediator. But this is not sufficient. It's required, but it's not sufficient to induce mitophagy. We need to induce mitophagy specifically only in the vicinity of damaged mitochondria, not fresh mitochondria that are just being generated. So this is happening through the pink parking pathway that selectively only activates DCT1, ubiquitinates DCT1 and activates mitophagy in the vicinity of damaged mitochondria. And that's how cells eliminate selectively damaged mitochondria. Now, in neurons, as I've told you this morning, mitophagy is playing an essential role in preventing neuronal death, preventing neurodegeneration. If mitophagy cannot function properly, then this accumulation of mitochondria eventually will cause neurodegeneration, will sensitize these neurons to neurodegeneration. So mitophagy is a protective mechanism in neurons that protects against neurodegeneration and contributes to long-term survival of these neurons. And I will finish by thanking my colleagues and collaborators at the University of Crete in Heraklion in Greece, because that's where I come from. These are the people who did the hard work, especially I have to thank two people, Kostas Palikaras. This is part of his PhD that he did. He's now an assistant professor at the Medical School of the University of Athens. And Irini Lionaki, a postdoc that did the biochemical characterization of mitophagy in C. elegans. Thank you very much for your attention, and if there are questions, of course, I would be happy to address them.